The stroke begins before the person feels it. A person can be walking through an ordinary day while the first stage of irreversible brain injury is already underway. There is no weakness yet, no slurred speech, no facial droop, and no reason to believe anything is wrong. But deep inside a narrow branch artery feeding the brain's white matter, blood flow has already fallen below the level required to sustain stable neuronal signaling. The stroke begins here, not when the body finally reveals it, but in the first invisible seconds when oxygen and glucose stop reaching tissue that depends on constant energy to stay alive. The earliest damage of a stroke is not the visible symptom. It is the sudden collapse of energy production inside neurons trapped downstream from the blocked vessel. The moment perfusion drops, mitochondria lose the oxygen and glucose required to generate ATP, the molecular currency that keeps every neuronal membrane stable. Without ATP, the sodium-potassium pumps embedded in the membrane begin slowing almost immediately. These pumps normally preserve the electrical difference between the inside and outside of the neuron, and once they begin failing, the cell loses the ability to protect its own internal environment. This is the real biological start of the stroke. Electrical instability before structural death. Sodium begins accumulating inside the neuron, while potassium leaks outward. Water follows sodium into the cell, causing early cytotoxic swelling that disrupts cellular architecture and weakens axonal conduction, and slows signal transmission through the white matter pathways that connect major cortical systems. The person still feels normal because behavior is a network event, but at the cellular level, the first neurons are already slipping into silence. This is what makes silent early stroke damage so dangerous. The brain can hide the first injury through collateral circulation and network redundancy. Nearby vessels may still deliver a weak trickle of blood around the blockage, and parallel white matter fibers may temporarily preserve speech, movement, balance, and conscious awareness. The outside world still sees a healthy person. The brain itself is already spending its reserve capacity to maintain that illusion. Around the future infarct core, the surrounding neurons enter the penumbra, a biologically unstable territory where the cells remain structurally alive but can no longer function normally. ATP levels continue falling, anaerobic metabolism begins to rise, lactate accumulates, and membrane pump failure spreads in waves across neighboring tissue. The person may still have no recognizable symptom, yet the brain is already dividing itself into two regions: neurons already progressing toward irreversible infarction, and neurons that are still temporarily salvageable if blood flow is restored fast enough. This is the most deceptive phase of stroke pathology because symptoms are not required for real damage. A tiny clot in a deep penetrating artery can begin killing neurons in the thalamus, basal ganglia, or frontal white matter without producing immediate paralysis or speech loss. The tissue is still undergoing the same ischemic chemistry: glutamate accumulation, calcium influx, mitochondrial stress, and progressive synaptic failure. The stroke is silent only to the person experiencing it. Inside the tissue, the damage is already fully real. The most dangerous consequence of this invisible beginning is that the first noticeable symptom may not belong to the first stroke at all. What finally becomes visible may be the delayed cost of this hidden injury: slower reaction speed, slight imbalance, reduced processing efficiency, or subtle memory hesitation that feels easy to dismiss as fatigue. By the time the person notices even these mild changes, the blocked vessel may already be reopened, while the neurons that crossed the survival threshold remain permanently gone. This is the pathology hidden inside the title of this episode. Stroke damage begins before symptoms, because symptoms belong to the failure of large-scale neural networks. While injury begins as a cellular energy crisis that starts within minutes of blocked blood flow, cells fail first, networks fail later. By the time the body finally reveals weakness, confusion, slowed cognition, or a larger second infarct, The earliest damage may already be permanently embedded inside the white matter architecture of the brain. The stroke, in biological terms, began long before the person ever knew it existed. What a silent stroke really is. A silent stroke is not a harmless event. It is most often a true small ischemic infarct or lacunar tissue injury that forms without the dramatic early warning signs people expect from a stroke emergency. The vessel still blocks, blood flow still falls, oxygen and glucose still disappear from the downstream tissue, and a small territory of neurons still crosses into irreversible damage. The only difference is that the injured region may be small enough, deep enough, or strategically placed in a way that allows the surrounding brain to temporarily hide the loss. The word silent creates the illusion that nothing serious happened, but the biology inside the tissue is exactly the same as any recognized ischemic stroke. ATP collapses, membrane pumps fail, sodium and water accumulate inside neurons. Glutamate begins building in the extracellular space, and calcium floods into vulnerable cells. The neurons do not care whether the person notices the event. A silent infarct still leaves behind permanent tissue loss, disrupted axonal pathways, and a lasting reduction in network efficiency that can shape cognition months or years later. Most silent strokes begin in the tiny penetrating arteries that supply the deep white matter, thalamus, and basal ganglia. These small vessels are especially vulnerable to years of hypertension, diabetes, endothelial dysfunction, and vessel wall thickening. Over time, the lumen narrows until one branch finally closes completely. Because the affected region is often small, surrounding pathways can reroute around the damage just well enough to preserve outwardly normal speech, movement, and awareness. This creates one of the most dangerous illusions in vascular neurology. The brain can look normal while a permanent lesion is already forming in secret. The most common structural result is the lacunar infarct, a small cavity left behind after a penetrating vessel loses flow and the downstream neurons die. Even though the lesion may be tiny, its location inside white matter communication highways makes it disproportionately important. These tracts connect frontal executive regions with memory systems, motor planning circuits, emotional regulation networks, and deeper relay centers. A tiny cavity in one of these pathways may not produce immediate collapse, but it can slow the speed and synchronization of signal transfer across the entire system. This is why many silent strokes are only discovered much later during MRI scans performed for memory changes, dizziness, slowed thinking, subtle balance issues, or after a larger visible stroke finally occurs. The scan may reveal scattered lacunes or old white matter infarcts that formed long before the first recognized symptom. What feels clinically new may actually be the delayed expression of vascular damage that has been accumulating silently for years. The most deceptive aspect of a silent stroke is that the first visible problem is often not the stroke itself, but the cumulative burden left behind. One small lesion may create almost no obvious dysfunction. Several distributed lesions begin slowing executive processing, reducing multitasking efficiency, weakening memory retrieval speed, and subtly destabilizing gait. By the time these changes become noticeable, the person may believe the issue began recently, when in reality, the biological process may have started with an invisible infarct months or even years earlier. This is where silent stroke becomes especially dangerous under the pathology of fear framework. The brain spends reserve capacity to conceal each lesion. Parallel fibers compensate, neighboring cortical systems absorb the load, and frontal subcortical loops reroute around the damaged tract. But every successful concealment consumes part of the brain's long-term resilience budget. The person feels normal, not because the injury was harmless, but because the network sacrificed future efficiency to protect present function. The deeper truth is that silent stroke is often the architectural beginning of future vascular cognitive decline. Each hidden infarct weakens white matter integrity, slows frontal subcortical communication, and reduces the reserve needed to survive the next vascular insult. What finally appears as memory slowing, poor decision speed, mental fatigue, or a larger symptomatic stroke may simply be the moment the brain can no longer hide the structural debt created by years of silent damage. The first minutes: ATP failure and ionic collapse. The first minutes of a stroke are defined by loss, not by symptoms. The moment a clot lodges inside a cerebral artery, the downstream neurons are abruptly cut off from oxygen and glucose, the two substrates required for continuous aerobic metabolism. Unlike muscle or liver tissue, neurons store almost no usable energy reserve. They depend on uninterrupted blood flow every second. This means the biological clock of irreversible injury starts immediately, long before the body produces any visible sign that anything is wrong. The first thing the neuron loses is ATP. Inside healthy tissue, mitochondria continuously convert oxygen and glucose into this molecular energy currency, powering the membrane systems that preserve cellular stability. The most critical of these are the sodium-potassium pumps, which constantly force sodium out of the neuron and potassium back in. This invisible exchange maintains the electrochemical gradient required for action potentials, synaptic timing, and white matter signal speed. The moment ATP production falls, these pumps begin slowing, and the membrane starts losing control over its own internal environment. As pump failure spreads, sodium accumulates inside the neuron, while potassium leaks into the extracellular space. This immediately destabilizes the membrane potential and drags the cell toward uncontrolled depolarization. Water follows sodium through osmotic pressure, causing cytotoxic swelling that distorts dendrites, compresses intracellular structures, and weakens axonal conduction. The neuron still exists, but the fidelity of its signaling begins degrading within minutes. In white matter pathways, this translates into slower transmission speed before complete signal loss, which is why early stroke damage can remain behaviorally invisible while conduction failure is already underway. The next stage is ionic collapse. Once enough neurons lose membrane stability, they begin undergoing uncontrolled depolarization waves that spread across adjacent tissue. Instead of precise signaling, the tissue experiences electrical failure. Synaptic vesicles release glutamate into the extracellular space, while surrounding astrocytes, now starved of ATP themselves, lose the ability to clear it efficiently. The extracellular environment becomes chemically hostile, and the original blocked vessel is no longer the only problem. The damaged neurons are now amplifying their own destruction. This is the beginning of excitotoxicity. Excess glutamate overstimulates receptors on neighboring neurons, forcing ion channels to remain open and allowing massive calcium influx into cells that may still have been structurally viable. The injury now expands beyond the original perfusion territory because the chemical environment itself is spreading damage outward. A small arterial blockage can therefore begin transforming into a larger zone of threatened tissue, not simply through lack of blood flow, but through the neuron's own failure chemistry. Calcium overload is where threatened neurons begin crossing from reversible dysfunction into active self-destruction. Under normal physiology, intracellular calcium is tightly regulated because even small shifts can alter enzyme systems, mitochondrial behavior, and membrane integrity. During ischemia, the calcium flood becomes overwhelming. Proteases begin cleaving structural proteins, phospholipases attack membrane lipids, and endonucleases start damaging nuclear repair systems. The neuron is no longer simply starving. It is biochemically dismantling itself from the inside. At the same time, mitochondria suffer a second catastrophic blow. Already deprived of oxygen and glucose, they are now overloaded with calcium and exposed to rising oxidative stress. Their membranes lose integrity. ATP synthesis collapses further, and the cell enters a vicious cycle where energy failure worsens. Calcium overload, while calcium overload destroys the remaining energy machinery. This loop is the true molecular signature of the first minutes of stroke injury. The tissue now begins separating into two biological territories. At the center lies the ischemic core, where TP depletion is so complete that rescue is no longer biologically realistic. Surrounding it is the evolving penumbra, where collateral blood flow still supports a thin margin of metabolic survival. The body may still show no outward sign of danger, but inside the brain, the border between permanently lost neurons and temporarily threatened neurons has already been drawn. This is why the visible symptom is often delayed. Symptoms require enough white matter fibers, cortical networks, or motor pathways to fail together before behavior changes. Cellular collapse begins far earlier. By the time the person notices slowed speech, slight confusion, reduced balance, or delayed thought speed, the first minutes of a TP failure, ionic collapse, glutamate toxicity, and calcium-driven self-destruction may already be complete. The tissue decision was made long before awareness ever had a chance to catch up. The penumbra, alive but failing. The most deceptive territory in stroke pathology is not the infarct core. It is the penumbra, the ring of tissue surrounding the blocked center where neurons are still structurally alive, but no longer functioning normally. Blood flow has fallen below the level required for stable signaling, yet remains just high enough to delay immediate cell death. This creates a suspended biological state between survival and destruction. The neurons still exist, but their ability to transmit information, preserve timing, and maintain membrane stability is already failing. The ischemic core and the penumbra are separated by survival threshold, not by anatomy alone. In the core, a TP production has collapsed so severely that membrane integrity cannot be preserved, forcing rapid necrosis and irreversible tissue loss. In the penumbra, collateral vessels still deliver a reduced trickle of oxygen and glucose from neighboring arterial systems. This weak residual perfusion is enough to preserve structural integrity for a limited period, but not enough to sustain normal electrophysiology. The tissue is alive, but it is no longer healthy. This is the central illusion behind stroke damage before symptoms. A person may still move normally, speak clearly, and remain fully conscious because surrounding networks temporarily reroute around penumbral dysfunction. Parallel white matter tracts share the signal load. Adjacent cortical circuits absorb partial conduction delays, and frontal reserve capacity hides the earliest failures. But compensation at the network level does not reverse the chemistry inside the threatened tissue. ATP remains critically reduced, ion gradients remain unstable, and every minute of reduced perfusion pushes more neurons toward irreversible collapse. Function fails before structure because synaptic transmission is one of the most energy-sensitive processes in the brain. Even a moderate ATP deficit slows vesicle recycling, weakens action potential propagation, and reduces neurotransmitter clearance efficiency. White matter tracts begin losing synchronization, signal timing drifts, and cortical integration becomes subtly less precise. In real life, this may first appear as slower reaction speed, a momentary word-finding hesitation, a brief lapse in focus, or a barely noticeable shift in balance. These changes are often too small to be recognized as stroke, even though they directly reflect unstable penumbral tissue. In daily life, this may feel like taking slightly longer to answer a simple question, hesitating before stepping off a curb, or briefly losing the thread of a conversation before recovering. The survival of the penumbra depends on collateral circulation, one of the most important determinants of stroke outcome. These alternate vascular routes partially bypass the blocked artery and feed the threatened zone for nearby arterial territories. Strong collaterals can keep penumbral tissue viable for hours, extending the rescue window and preserving network function longer than expected. Weak collaterals collapse quickly, allowing the infarct core to expand outward in concentric waves. This makes the penumbra the true battlefield of ischemic disease, because it is the tissue that still has a biological future, but only temporarily. The penumbra is not healthy tissue waiting peacefully for rescue. It is already pathologic. Calcium influx continues at low levels. Glutamate remains elevated in the extracellular space. Lactate keeps accumulating from anaerobic metabolism, and mitochondrial stress progressively worsens. The neurons remain alive only because the residual blood flow has slowed the collapse, not because the collapse has stopped. This is why time is biologically destructive, even when the person still appears normal. As minutes pass, the border between penumbra and core begins shifting outward. This is infarct progression, the moment when previously salvageable neurons lose the final perfusion margin required to remain structurally intact. What begins as reversible electrical failure slowly hardens into membrane rupture, organelle destruction, and permanent tissue loss. In visual terms, the dim, unstable ring around the clot gradually darkens into the same irreversible absence as the central core. This is why the first visible symptom may still be delayed. If the penumbra initially involves deep white matter tracts with strong redundancy, behavior can remain deceptively normal while the lesion expands microscopically. Only when enough fibers lose conduction together does the system-level failure become obvious. A delayed weakness, sudden word-finding pause, transient gait drift, or abrupt slowing in thought may simply mark the moment the penumbra crossed the network's compensation threshold. The symptom does not reveal when the stroke began. It reveals when compensation finally failed. The deeper fear hidden in this phase is that the most dangerous damage is often not the instantly dead tissue, but the tissue that remains alive long enough to stay invisible. The penumbra is the zone where the brain still has a chance to survive, yet also the zone where time silently converts function into permanent absence. By the time the body finally reveals the truth, part of what could once have been rescued may already be permanently embedded as old infarcted tissue inside the white matter architecture. Why some strokes stay invisible? Not every stroke announces itself through collapse, slurred speech, or sudden paralysis. Some infarcts remain clinically invisible because the damaged tissue lies in regions where the brain can temporarily absorb the loss without immediate behavioral failure. The vascular event is still real, the neurons are still dying, and the white matter architecture is still being permanently altered. But the location of the lesion determines whether the network can hide the damage long enough to delay awareness. The most common invisible strokes occur in the deep white matter tracts. These pathways function as the communication highways linking frontal executive regions, motor planning systems, memory networks, and subcortical relay centers. A small infarct in one tract may not immediately disable movement or speech because parallel fibers can partially reroute around the disruption. The signal still moves, but less efficiently. This creates the dangerous illusion of normality, while microscopic disconnection has already begun. The thalamus is another common site of clinically silent injury. Because it acts as a relay center for sensory integration, attention regulation, cortical timing, and arousal, tiny lesions in less dominant nuclei may initially produce effects too subtle to trigger alarm. Instead of dramatic numbness or altered consciousness, the earliest manifestations may be slightly delayed attention shifts, reduced processing speed, mild sleep disruption, or a vague sense of mental heaviness that feels easy to dismiss as fatigue. Silent infarcts in the basal ganglia are equally deceptive. These structures regulate movement timing, habit loops, task initiation, and cognitive motor efficiency. A tiny lesion here may spare gross strength while subtly degrading fluidity, reaction speed, and the automatic feel of movement. The person still walks, still uses their hands, and still performs daily routines, but transitions feel fractionally slower and mentally heavier. What seems like ordinary tiredness may actually be the delayed network expression of a hidden infarct. In ordinary routines, this can feel like taking longer to get dressed, needing more effort to type smoothly, or finding that simple multitasking suddenly feels mentally exhausting. Lesions in the non-dominant hemisphere can remain hidden even longer. Small right-sided subcortical or parietal infarcts may spare language and major strength pathways, allowing the most recognizable public stroke symptoms to remain absent. Yet these same lesions can still weaken visuospatial integration, divided attention, emotional regulation, and body awareness in ways too mild to be immediately interpreted as neurological injury. The person may simply describe feeling off without understanding that a permanent structural lesion already exists. Another reason strokes remain invisible is delayed symptom emergence. Not every lesion reaches network failure threshold immediately. In some cases, conduction slowing, evolving edema, delayed edema, and secondary inflammatory signaling, and progressive axonal dysfunction gradually expose the damage over hours or even days. The first day may feel normal. By the second, focus feels slower. By the third, memory retrieval feels slightly delayed. This disconnects the visible symptom from the vascular event, making recognition even less likely. The brain's reserve capacity plays a critical role in this concealment. Younger brains or individuals with stronger baseline white matter integrity may tolerate a surprising amount of subcortical injury before visible dysfunction appears. Neighboring pathways absorb the load, frontal systems reroute around the lesion, and cortical redundancy masks the deficit. But every successful concealment consumes part of the brain's long-term resilience budget. The injury is not harmless. The cost is simply deferred into the future. This is why the first visible decline is often cumulative rather than singular. One silent lacune in frontal white matter may produce almost no obvious dysfunction. Several distributed lesions begin slowing executive processing, weakening task switching, reducing memory retrieval speed, and subtly destabilizing gait. By the time the symptoms become undeniable, the network may already contain years of invisible structural scars that have been slowly eroding reserve capacity. The deeper fear in this chapter is that the absence of classic stroke symptoms does not mean the absence of real damage. Invisible infarcts are often the biological foundation of vascular cognitive impairment, mood dysregulation, gait instability, and later major stroke vulnerability. The brain can lie for a while, but every hidden infarct leaves behind structural debt. What finally appears as memory slowing, slower thinking, imbalance, or a larger visible stroke may simply be the moment the network can no longer hide the accumulated cost. Damage after blood flow returns. The return of blood flow is not always the end of the stroke. In many cases, it marks the beginning of a second injury phase that unfolds inside tissue already weakened by a TP failure, ionic collapse, and mitochondrial stress. Once the blocked vessel partially reopens or collateral circulation improves, oxygenated blood re-enters neurons that survived the first wave only because they were suspended inside the unstable penumbra. At first, this seems like rescue, but inside metabolically damaged cells, reperfusion can trigger a new cascade that continues expanding permanent injury. The central problem begins inside the mitochondria. During the ischemic phase, these organelles were deprived of oxygen, overloaded with calcium, and pushed to the edge of collapse. When oxygen suddenly returns, they do not instantly resume healthy ATP production. Instead, damaged electron transport chains begin leaking unstable electrons that react with oxygen to form reactive oxygen species. These highly aggressive molecules attack membrane lipids, mitochondrial proteins, and nuclear repair systems, converting partial survival into molecular self-destruction. This oxidative burst is especially dangerous in neurons because their membranes are rich in delicate lipids that are essential for receptor stability, ion channel behavior, and synaptic signaling. Once these lipids begin oxidizing, the membrane becomes more permeable, less selective, and increasingly unstable. Neurons that survived the original ATP crisis can now become structurally doomed hours after the blocked vessel has reopened. The clot may be gone, Yet in vulnerable tissue, the chemistry of injury can continue. The vascular lining itself also enters a second failure phase. Endothelial cells that form the blood-brain barrier were already weakened by ischemia, ATP depletion, and inflammatory signaling. When reperfusion floods the region with oxygen radicals and circulating inflammatory mediators, the tight junction proteins that normally seal these vessels together begin to fail. The barrier becomes porous, allowing plasma proteins, fluid, and signaling molecules to leak into the surrounding extracellular space. This is the beginning of vasogenic edema, a more dangerous form of swelling than the early cytotoxic phase. The tissue is no longer swelling only because neurons are pulling in water. It is now swelling because the vascular compartment itself is leaking into the brain. White matter tracts become mechanically compressed, capillary flow is squeezed by rising pressure, and microvascular perfusion begins worsening again, despite the fact that the original vessel may already be reopened. At this point, the stroke is no longer just a blocked blood flow problem. It becomes a compartment failure problem. The tissue space expands, signal highways inside the white matter lose physical integrity, and the pressure from edema further compromises surrounding capillaries. This means blood flow restoration can paradoxically create new zones of threatened tissue by compressing the microcirculation around the lesion border. Inflammation now becomes the dominant mechanism of delayed damage. Activated microglia, infiltrating immune cells, and vascular inflammatory mediators begin releasing cytokines, proteases, and additional oxidative molecules into the reperfused region. The original ischemic event created the first wave of excitotoxic injury. This inflammatory phase now expands the border through delayed apoptosis, axonal degeneration, and progressive white matter destabilization. The visible clinical worsening that may appear hours later often reflects this second wave expansion rather than the original clot. White matter is especially vulnerable during this delayed phase. Axons that initially survived on reduced perfusion can still lose conduction precision as oligodendrocyte support weakens, myelin integrity deteriorates, and axonal transport systems fail. The result is slower processing speed, less synchronized frontal subcortical communication, and the subtle cognitive slowing that may not appear until days after the vascular event. The person may feel stable at first, only to notice that thought speed, balance, or working memory begins worsening later. This is the deeper illusion hidden in reperfusion injury. Restored blood flow does not rewind time. The tissue remembers the ischemic phase. Mitochondria remain damaged, membranes remain unstable, the blood-brain barrier remains fragile, and inflammatory chemistry continues rewriting the lesion border long after the original vessel problem appears solved. The visible symptom may still be delayed, but the biology of permanent injury is still actively progressing. The most dangerous consequence of this second-wave damage is that it consumes what remained of the penumbra. Tissue that initially survived on collateral flow may cross into irreversible death, not because blood flow stayed blocked, but because restored circulation arrived too late for a biologically unstable neuron to recover safely. By the time the person feels normal again, the final borders of permanent white matter loss may still be expanding in silence. When silent damage becomes memory loss. The first visible consequence of silent stroke damage is often not paralysis, speech loss, or sudden collapse. It is cognitive slowing. Deep white matter infarcts gradually interrupt the communication highways that connect the frontal cortex to memory systems, motor planning networks, emotional regulation circuits, and subcortical relay centers. The result is not immediate disability, but reduced efficiency across the systems that make fast thinking, smooth decisions, and effortless multitasking possible. The person may still function independently, while the brain is already operating with less speed and less reserve. The earliest change is usually processing speed. Tasks that once felt automatic begin requiring more deliberate mental effort. Conversations are still understandable, but responses arrive a fraction slower. Switching between tasks becomes less fluid, decision-making feels heavier, and complex information takes longer to organize into action. This is the delayed behavioral expression of old white matter lesions that have already weakened the synchronization between frontal executive systems and deeper relay pathways. Frontal subcortical loops are especially vulnerable to this kind of silent damage. These circuits depend on fast, precisely timed signal transfer through intact white matter tracts. When multiple silent lacunes interrupt these routes, the brain begins losing speed before it loses intelligence. The person still understands the task, still knows the right answer, and still recognizes the goal. But the neural route to execute that thought becomes slower and more energy demanding. Memory changes often emerge next, especially when silent infarcts involve thalamic relay systems or the frontal white matter pathways that support retrieval. The problem is not always storage of memory itself, but access speed. Names take longer to surface, recent details require more effort to retrieve, and working memory becomes easier to overload during information-heavy situations. What feels like harmless forgetfulness may actually reflect a structural slowdown in the pathways that connect frontal retrieval systems with deeper memory networks. This is often the stage where someone notices they reread the same sentence twice, lose their place during a familiar routine, or need extra seconds to recall a name they normally know instantly. This is where silent stroke pathology becomes especially deceptive. The person may interpret the changes as stress, aging, poor sleep, or mental fatigue because the symptoms emerge gradually rather than dramatically. But under the surface, the real issue is disconnection. Multiple microscopic lesions have begun slowing the timing precision of distributed neural systems, forcing the brain to spend more energy to produce the same mental output. Gait and balance often begin changing alongside cognition because the same white matter highways that support executive speed also coordinate movement timing and spatial adjustments. Walking becomes less automatic. Turning may require more conscious control. Steps feel fractionally less fluid, especially when multitasking or moving through visually complex environments. The person may describe this as feeling cautious, slower, or less confident, not realizing that the same silent lesions affecting cognition are also disrupting motor timing networks. Mood and emotional regulation can also shift as frontal subcortical circuits lose efficiency. The person may become more mentally fatigued, more easily overwhelmed by decisions, or less adaptable under cognitive stress. These changes are often subtle enough to be interpreted as personality shifts or burnout, when in reality, they may reflect the delayed network consequences of deep vascular injury that was never recognized when it first occurred. The most dangerous part of this phase is that the visible decline often feels disconnected from the original stroke event. The infarct may have happened months or years earlier. By the time the person notices slower thinking, memory hesitation, reduced multitasking, or gait instability, the original lesion has already hardened into old white matter damage. What appears as a new symptom is often simply the first moment the accumulated burden becomes large enough to cross the brain's compensation threshold. This is how silent infarcts evolve into vascular cognitive impairment. The decline does not begin when the first lesion forms. It begins when enough hidden lesions have weakened the frontal subcortical communication system so extensively that the brain can no longer maintain the illusion of normal speed. The person may still look healthy, still speak clearly, and still live independently. But the network is now operating on a reduced margin of efficiency that leaves it dangerously vulnerable to the next vascular insult. The deeper fear in this stage is that memory loss and slowed thinking are often not the beginning of disease. They are the delayed visibility of structural damage that began long before symptoms ever existed. By the time the person notices the change, the biological architecture of the decline may already be permanently embedded inside the white matter pathways that once kept thought fast, stable, and effortless. The point of no return. The true point of no return is rarely the first clot. It is the moment the brain can no longer compensate for the structural debt created by multiple silent injuries. For a long time, neighboring pathways reroute signals around damaged tracts. Frontal systems absorb timing delays, and reserve capacity hides the earliest deficits. But every hidden infarct consumes part of that reserve. The brain can protect function only as long as enough intact white matter remains to sustain synchronized communication between major networks. At first, compensation is remarkably effective. One lacune in frontal white matter may create almost no visible consequence. A second lesion in the thalamic relay system may only slightly slow retrieval speed. Another small infarct in the basal ganglia may reduce movement fluidity by a margin too small to notice. But these lesions do not remain isolated in their effect. Each one forces neighboring pathways to carry more signal load, increases conduction distance, and places higher metabolic demand on fibers that were never meant to absorb permanent detours. This is where large-scale white matter disconnection begins. The communication highways linking executive planning, working memory, motor timing, emotional regulation, and attention control start losing the synchronization required for efficient cognition. Signals still move, but they arrive later, less precisely, and with greater energy cost. The person experiences this as sudden decline, but biologically, it is the end point of years of silent rerouting and progressive reserve depletion. Once enough tracts are compromised, frontal subcortical loops begin failing as systems rather than as isolated fibers. Decision speed collapses, task switching becomes unreliable, working memory becomes unstable, and gait timing begins breaking under dual task conditions. This is the threshold where the brain can no longer spend reserve capacity to preserve normal behavior, because the reserve itself has already been structurally consumed. The most deceptive part of this stage is the feeling of suddenness. A person may believe their cognition worsened abruptly over weeks or months, when the biological process may have been progressing invisibly for years. The visible decline simply marks the moment the lesion burden exceeded the network's ability to conceal it. The first obvious symptom is not the first meaningful injury. It is the first failure the brain could no longer hide. This threshold also amplifies future vulnerability. Once large-scale disconnection is present, even a relatively small new infarct can produce disproportionately severe dysfunction. A lesion that would once have remained silent may now trigger obvious memory failure, executive breakdown, or major gait instability because the remaining reserve margin is too thin to absorb any additional loss. Silent damage transforms the brain into a system that becomes progressively easier to break. The white matter itself now enters a self-reinforcing decline loop. Reduced network efficiency leads to lower cognitive activity, weaker synaptic maintenance, less adaptive plasticity, and poorer utilization of compensatory pathways. At the same time, chronic vascular risk factors continue damaging the same microvessels that produced the earlier lesions. The brain is no longer just carrying old scars. It is operating inside a biologically unstable environment that makes every future injury more likely and more visible. This is why the point of no return is best understood as network collapse, not simply tissue death. Individual neurons may have died months or years earlier, but the true irreversible threshold is crossed when the architecture of compensation itself fails. Once the brain loses enough reserve to reroute around silent infarcts, decline can accelerate into persistent functional impairment despite partial compensatory plasticity. The deeper fear inside this phase is that the visible symptom forcing recognition may arrive only after the most important protective system is already gone. The person notices slower decisions, lost mental sharpness, unstable gait, or growing dependence in complex tasks. But the real event happened earlier. The network lost the structural flexibility that once allowed it to lie. From this point forward, every additional lesion carries exponentially greater consequences because the brain is no longer failing silently. it is failing in public. Outro. The next stroke is bigger. The most dangerous stroke is often not the one that announces itself through sudden collapse. It is the one that begins in silence, reshapes the brain in secret, and waits until compensation fails before revealing what was already lost. By the time a person notices slowed thinking, subtle imbalance, delayed memory retrieval, or the first unmistakable weakness, the earliest injury may already be permanently embedded inside the white matter architecture that once kept the brain fast, synchronized, and resilient. Blocked blood flow can damage neurons within minutes, destabilize penumbral tissue for hours, and continue expanding permanent injury through delayed oxidative stress, edema, and inflammatory remodeling long after the original vessel problem appears solved. What looks like a normal day on the outside may already contain the first irreversible shift in the brain's internal structure. The danger was never defined by when the symptom appeared. It was defined by when the neurons first lost the energy required to survive. This is why silent infarcts are so dangerous under the pathology of fear framework. They do not simply leave behind isolated scars. They weaken the reserve systems that would have protected the brain from the next vascular insult. Every hidden lesion reduces white matter integrity, slows frontal subcortical communication, and narrows the margin of safety that once allowed surrounding pathways to compensate. The brain may continue functioning normally for a while, but each concealed injury makes the next event more likely to become visible, larger, and more destructive. The next stroke is often bigger, not only because the vascular risk factors remain active, but because the network itself has already been pre-weakened. A new clot does not land on a healthy communication system. It lands on white matter tracks already slowed by old lacunes, relay centers already burdened by delayed conduction, and frontal circuits already operating with reduced reserve. A lesion that once might have remained silent can now trigger immediate executive failure, memory collapse, major gait instability, or full symptomatic stroke presentation. This is the hidden escalation pathway of silent stroke damage. The first event spends reserve. The second event exposes the cost. By the time the larger stroke finally announces itself, the true beginning of the disease may already be months or years behind the person's awareness. What feels like a sudden catastrophe is often the visible endpoint of a long, invisible chain of vascular injuries that slowly rewrote the architecture of the brain. The deeper fear is that the brain rarely announces the first injury. It announces the moment it can no longer hide the accumulated damage. The visible stroke, the sudden weakness, the slowed speech, or the collapse in memory may simply be the first public sign that the internal reserve systems were already exhausted by silent infarcts that no one ever knew existed. And this is what makes blocked blood flow before symptoms so biologically devastating. The damage begins at the cellular level, spreads through white matter architecture, consumes network reserve, and waits until the next lesion arrives to reveal how much was already lost. By the time the body finally forces recognition, the real beginning of the stroke may already belong to the past, permanently sealed inside the pathways that once made thought, movement, and memory feel effortless. The first stroke is often never the one the person remembers. The brain can spend years hiding the damage, rerouting around dead tissue, borrowing time from reserve pathways, and masking the slow collapse of its own communication system. But every silent infarct leaves behind structural debt, and the moment that debt can no longer be paid, the illusion of normality ends all at once. What finally appears as weakness, slowed speech, memory collapse, or a larger visible stroke is often not the beginning of the disease. It is the first moment the brain loses the ability to protect the person from what has already been happening in silence. The visible symptom is only the public version of a cellular disaster that may have started years earlier. By the time the stroke is finally seen, the real damage may already belong to the past. The most dangerous injury was the one that began before the person ever felt it, spreading through white matter pathways, consuming reserve, and waiting for the next lesion to expose what was already lost. This is pathology of fear, and this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.